So good afternoon to all. So in the last class already we have discussed the process of gametogenesis. So in that gametogenesis there are two types. One is spermatogenesis, another one is the oogenesis. So spermatogenesis process already we have discussed and already we have completed. The next part is the next type of process is oogenesis. So that today we'll discuss. So first we should know what is oogenesis. So oogenesis means formation and maturation of the egg or the ovum. That is called as oogenesis. And where it is taking place? It is taking place in the female gonad, that is ovary. So complete definition is oogenesis is a process of formation and maturation of egg or the ovum within the ovary. So it is called as oogenesis. Okay, this complete definition you have to remember. And each ovary, a pair of ovary is present in the female, a pair of ovary. Each ovary is an irregular, that is a sac-like structure that is lined by the germinal epithelium. Some of the cells of the germinal epithelium they divide and differentiate and they give rise to the egg or the ovum. So epithelium is there, that is the germinal epithelium is there. So though some of the cells of germinal epithelium divide and they go and differentiate to give rise to the egg or the ovum. So the process of oogenesis is very similar to the process of spermatogenesis, and it is also involves the three phases. So like that in the spermatogenesis we have seen, that is phase of multiplication and the phase of growth and the phase of maturation. So these three phases are same, the names are same as we have seen in the spermatogenesis. So one by one we will discuss the phase of multiplication. Here during this phase some of the cells, which cells? The germinal cells or germinal epithelial cells, they divide repeatedly, go on repeatedly they are go on dividing by the process of cell division, that is mitotic division, and they produce the large number of cells or diploid daughter cells, and these diploid daughter cells are called as oogonia. ಅಲ್ಲಿ ಏನು ಕರಿತಿದ್ವಿ ನಾವು ಸ್ಪರ್ಮಟೋಜೆನೆಸಿಸ್ನಾಗ ಸ್ಪರ್ಮ್ಯಾಟೋಗೋನಿಯಾ ಅಂತ ಕರಿತಿದ್ವಿ. ಹಿಯರ್ ವಿ ಶುಡ್ ಕಾಲ್ ದಿ oogonia. So same similar things here also, that during this process what happens, the germinal cells, germinal epithelial cells, they go on dividing 2 4 8 like that, they go on repeatedly dividing by the process of mitosis, and they produce large number of diploid daughter cells, they are called as oogonial cells or oogonia. So some of the oogonia continue with the division, while some of them enter into the next phase called as the phase of growth. So type A, type B spermatogonia ಅಂತ ಹೇಳಿದ್ವಲ್ಲ ಅಲ್ಲಿ, so type B enter into the growth phase. So here also same, some oogonia continue to divide while some are entering into the next phase, that is called as phase of growth. So in the next slide you can see the phase of growth. The growth phase in the oogenesis is much prolonged than the spermatogenesis and is differentiated into pre-vitelline phase and the vitelline phase. So as compared to the spermatogenesis, here there is a little difference, that is here pre-vitelline phase and vitelline phase, there are two phases we are going to see. So the pre-vitelline phase is characterized by the nuclear and cytoplasmic changes. Here what happens in the pre-vitelline? Vitellogenesis you heard. Vitellogenesis means the process of formation of yolk, so that is called as vitellogenesis. Here the pre-vitelline phase, so it is characterized by, or the main feature of this pre-vitelline phase is by the nuclear and cytoplasmic changes ಅಂದರೆ here the cytoplasmic changes are taking place and the nuclear changes are taking place in the pre-vitelline phase. And the vitelline phase, what happens, and this phase is characterized by deposition of large number of reserve food material in the form of yolk. Vitelline phase means vitellogenesis, ಇಲ್ಲಿ ಡಿಪಾಸಿಷನ್ ಆಫ್ ಯೋಕ್ ಆಗ್ತದೆ. ಸ್ಪರ್ಮೆಟೋಜೆನೆಸಿಸ್ ಹೆಂಗಿರ್ತಲ್ಲ ಇಲ್ಲಿ vitellogenesis ಅಂದರೆ there is no vitellogenesis in the males. So here in the females there is a egg is there, no? That's why there is a yolk deposition takes place. So in the vitelline phase it is characterized by the deposition of the large amount of reserve food materials. So those reserve food materials are in the form of yolk or deuteroplasm. So as a result of these, oogonia grows enormously and is called as primary oocyte. So here what happens in the growth phase, the oogonia becomes enlarged or they grow. So oogonia grows enormously and this is called as primary oocyte. Okay, in the next slide, the phase of maturation. Phase of maturation. So during this phase, primary oocyte undergoes meiotic division. So the first meiotic division is a reductional division. Here the diploid primary oocyte divides into two haploid nuclei, but the cytoplasm divides unequally. So here in the maturation phase, what happens during this phase, the primary oocyte, so that is a growth phase. We have seen in the growth phase the oogonia undergo growth, and that growth oogonial cell is called as primary oocyte. That primary oocyte undergoes the meiotic division. So the first meiotic division is a reductional division, already I have told in the spermatogenesis process also. Here diploid primary oocyte divides into two haploid nuclei, but the cytoplasm divides unequally. The two haploid nuclei are going to be formed and cytoplasm divides unequally. So this resulting in the formation of a large haploid secondary oocyte. What is formation? What formation takes place here? Formation of large haploid secondary oocyte and a small haploid polar body. So they enter into the second meiotic division. They again enter into the second meiotic division. So at the time of second meiotic division, which is a equational division, so the secondary oocyte as well as the polar body divides again. Here what happens, the polar body also divide and secondary meiotic division ಆಗ್ತದೆ. ಇಲ್ಲಿ ಅಲ್ಲಿ ಏನಾಗ್ತದೆ, equational division. Equatorial ಅಂದರೆ ಸೇಮ್ ಈಕ್ವಲ್ ಡಿವಿಷನ್ ಆಗ್ತದೆ. ದ ಸೆಕೆಂಡ್ರಿ oocyte ಡಿವೈಡ್ ಆಗ್ತದೆ ಆಸ್ ವೆಲ್ ಆಸ್ ದ ಪೋಲಾರ್ ಬಾಡಿನೂ ಡಿವೈಡ್ ಆಗ್ತದೆ. ಸೊ ನೆಕ್ಸ್ಟ್ ಐ ವಿಲ್ ಶೋ ಇನ್ ದಿ ಸ್ಕಿಮ್ಯಾಟಿಕ್ ರಿಪ್ರೆಸೆಂಟೇಷನ್. ಸೊ ದ ಫಸ್ಟ್ polar body divides equally. The first polar body already it has been formed, so that polar body divides equally and gives rise to two polar bodies. Again that polar body gives rise to two polar bodies, whereas the secondary oocyte divides unequally and producing large ootid. So that already large ootid is formed and a small second polar body or polocyte unequally producing large ootid and a small second polar body is going to be formed. So the ootid again develops into an egg. ಮುಂದೆ ಏನಾಗ್ತದೆ, ooted ಡೆವಲಪ್ ಆಗಿ ಎಗ್ ಆಗ್ತದೆ, ವೇರಾಸ್ polar bodies disintegrate or sometimes disgenerate or disappear. So thus at the end of the oogenesis process, 4 cells are formed. So out of which there is a single functional ooted and 3 non-functional polar bodies are formed. Totally 4 cells are formed. One is functional, that is called as ooted, and another remaining 3 are non-functional, those are polar bodies. The polar bodies disappear ಆಗ್ತಾ ಇರ್ತಾವೆ, ಅದಕ್ಕೆ ಡಿಸ್ಇಂಟಿಗ್ರೇಟ್ ಆರ್ ಡಿಸಪಿಯರ್ ಆಗ್ತಾ ಇರ್ತಾವೆ ಬಿಕಾಸ್ ಆಫ್ ದ ಲ್ಯಾಕ್ ಆಫ್ cytoplasm. So this is the oogenesis process, and you can see clearly in the next slide, that is the schematic representation. The first one is the germinal cells or primary germ cells with a diploid number of chromosomes, and this undergoes the mitotic division. Again the mitotic division or that process is called as multiplication phase, and those cells enter into the growth phase, and some cells will enter the growth phase, and those cells can grow in size, and that is called as primary oocyte, and this is a growth phase. And again the primary growth phase entered into the maturation phase. So here in the growth phase, the first maturation division taking place, one unequal division, one is a larger one cell and another one is the smaller cells. The smaller cell is called as the polar body, and the larger one is called a secondary oocyte. Again the secondary oocyte undergo the unequal division. One is a larger cell, another one is the smaller cell. The smaller cell is called as second polar body, and the larger one is called as the mature ovum. And the smaller from the primary spermatocyte there is a small cell is formed, that is a polar body, and that polar body again divides into two small equal cells called as polar bodies. So at the end of this process, so three polar bodies and one larger one, that is called as ooted. So one is functional ovum is going to be formed, and three polar bodies are formed, and these polar bodies are disappeared at the end due to the lack of the cytoplasm. Okay, so this is the process of oogenesis. So this may be asked for 5 marks or sometimes 10 marks. So if the process is asked for only one, that is single process, oogenesis process, with the schematic representation, explain the process of oogenesis, means 5 marks or sometimes 10 marks also. Like that spermatogenesis also. So sometimes both, gametogenesis they may ask, the process of gametogenesis. So then you have to write both the answers. Okay, if there is any doubts you can ask me. You have any doubts or not? No doubts? Can I ask how many phases are seen in the oogenesis process? How much? 3. Very good. Which are those? Phase of multiplication, then phase of growth, and phase of maturation. So these 3 phases we can see in the oogenesis process. Okay, so what is oogenesis? Tell me. Oh, right, good. Oogenesis is the process of formation of what? Mature egg, where it is going to be formed in the female gonad, that is the ovary, isn't it? Okay, good. So if there is no doubts, we can end up this session, and in the next class we are going to discuss the another part, that is the I think this has been completed. So a small part is remaining, that is the structure of spermatozoa. Structure of spermatozoa. So sometimes they may be asked or they may not be asked, but it is a part of the spermatogenesis. So that's why you have to study the structure of spermatozoa. We can study in the next class, otherwise we can finish up today only. Okay, we'll finish today only. We'll finish today only. Structure of spermatozoa. So this is the structure of sperm. Sometimes they may be asked. They may ask for 5 marks. So that's why it is also necessary to explain the structure of sperm. Without knowing the structure of sperm, we cannot understand the process of spermatogenesis. So many times the structure of spermatozoa is asked for 5 marks. So this is the complete structure of the sperm. So you can see here main 3 parts, that is the head, neck, and tail. So and there is some middle part that is called as middle piece. Head, neck, middle piece, and the tail. Here there is a structure, and there is a cap-like structure is there, no, in the head region, that is called as acrosome, and in the middle there is a granules are there, that is a granules like structure called as nucleus present in that, and that contains chromosomal material, and last membrane that covers the acrosome is called a plasma membrane, and in the middle piece there are a number of mitochondria are present. Those are the energy sources for swimming or motility of the sperm. Okay, one by one we can study. So here the structure of sperm. A mature human sperm is highly specialized sex cell and measuring about 52 to 62 micron in length. Okay, the measuring on the how much it is, the measurement of the sperm 52 to 62 microns. It is not visible from the naked eyes. We cannot see from the naked eyes. We require a microscope for the mortality, or if we want to observe the sperm means. So it is differentiated into four distinct regions. Already I have told: head, neck, middle piece, and the tail. So first one is head. The head is slightly flattened as we saw in the diagram, and pear shaped, slightly flat like structure, and it is about 4 to 5 micron in length. How much the length is? 4 to 5 micron in length, and 2.5 to 3.5 micron in width. The width and the length, how much it is, that you have to remember. Then it consists of mainly two parts, namely two parts, that is in the head region there are two parts we can see. One is the nucleus and another one is the acrosome. Already I have shown in the diagram. So what is nucleus and what is the acrosome? And the nucleus is large and occupies the major portion of the head. Means that in the two parts, and the acrosome and the nucleus, the nucleus occupies the larger portion of the head, and it contains haploid set of paternal chromosome. Paternal, maternal means mother side, the paternal means the father side, that is the male side, male genetic material it consists. So the acrosome or Galea capitis, that is we can call it as acrosome, otherwise Galea capitis, is present anterior to the sperm nucleus. And anterior to the sperm nucleus means in the anterior portion of the nucleus or the head we can find the acrosome, that is a cap like structure. So that is called as acrosome, that is the head part. Next slide. In the next slide, it forms a cap like structure or cap like covering at the anterior 2/3 of the nucleus, and it is derived from the Golgi apparatus. Golgi apparatus, from where it is derived. Acrosome is derived from the Golgi apparatus, and that containing several granules, which contains the hydrolytic enzymes like hyaluronidase and acrosin and neuraminidase. So these are the hydrolytic enzymes which are present in the nucleus. So nucleus are where? In the nucleus or the acrosome. So acrosome is derived from where? It is derived from the Golgi apparatus, and that containing granules, hydrolytic enzymes, that may be acrosin, hyaluronidase and neuraminidase. So these enzymes which are capable of dissolving the tissue. Why these are required? These are involved in the dissolving the tissue and helps the sperm in penetrating the egg during the fertilization. At the time of fertilization, for the penetration purpose, to the egg, and the sperm is going to be penetrate the egg. So at the time of fertilization process, so at that time we need these enzymes. Okay, this is about the head. Next, second part is the neck. The neck is short and a constricted region of the sperm, measuring about 0.3 microns in the length and present between the head and the ಮಿಡಲ್ ಪೀಸ್ ಅಂದರೆ ಹೆಡ್ಡು ಮತ್ತು ಮಿಡಲ್ ಪೀಸ್ ನಡುವೇನು ಒಂದು ಕನ್ಸ್ಟ್ರಿಕ್ಟೆಡ್ ಪಾರ್ಟ್ ಇರ್ತದೆ ಓನ್ಲಿ 0.3 ಮೈಕ್ರಾನ್ ಇರ್ತದೆ ಲೆಂತ್. ಸೊ ಅದಕ್ಕೆ ಏನಂತ ಕರಿತೀವಿ, ಶಾರ್ಟ್ ಪಾರ್ಟ್ ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಎ ನೆಕ್. ಆ್ಯಂಡ್ ದ ನೆಕ್ ಕಂಟೈನ್ಸ್ 2 centrioles. The centrioles are present in the neck region. Namely, there are 2 centrioles are named as proximal centrioles and the distal centrioles. What are those what are those centrioles? Proximals and distal centrioles. The proximal centriole lies just behind the nucleus, where it is present anta, where it is located. Proximal means it is located just behind the nucleus in its concavity. And behind the proximal centriole lies the another centriole, that is a distal centriole. And these arising from a distal centriole is a long fiber called as axoneme or the axial filament. Means the distal filament arising from a distal centriole arising from a long fiber called as axoneme or the axial filament. So next one is the middle piece. The middle piece is a broad region of the sperm and it is followed by the neck. Means the neck ಆದಮೇಲೆ ಯಾವ್ದು ಬರ್ತದೆ, ಮಿಡಲ್ ಪೀಸೇ ಬರ್ತದೆ. and it is present between the neck and the tail. So it has a large number of mitochondria, means it consists of mitochondria, act like an energy source, which are spirally coiled around the central axonema. They are in the form of spirally coiled structures. So the mitochondria provides energy for the motility of the sperm. So if the sperm has to move or the motility agಬೇಕಂದ್ರೆ it needs the energy. So that energy is provided by the mitochondria. So the middle piece has a very thin layer of the cytoplasm. ಅಂದರೆ ಮಿಡಲ್ ಪೀಸ್ ಹೆಂಗಿರ್ತಪ್ಪ ಅಂದ್ರೆ ವೆರಿ ಥಿನ್ ಲೇಯರ್ ಆಫ್ ದ cytoplasm and that is surrounded by the plasma membrane. So next the last part is tail. So tail is the longest region of the sperm body. The longest ಯಾವದಪ್ಪ ಅಂದ್ರೆ ಪಾರ್ಟು tail and is about 55 micron in the length. You can see the length of the tail. So 55 micron in length. It is differentiated into anterior main piece and the posterior end piece. There are 2 pieces or 2 parts are differentiated. One is anterior main piece we can call, another one is the posterior end piece. So the main piece contains the central axial filament. The main piece ಏನಿರ್ತಂದ್ರೆ that contains the central axial filament and an additional fibrous sheath. So this is surrounded by a thin film of cytoplasm and it has no mitochondria, and there is no mitochondria in the main piece. And the end piece is the last part of the spermatozoa and sperm tail. So it consists of axial filament and without being surrounded by the plasma membrane. So axial filament is there but it is not surrounded by the plasma membrane. So because of the vibratile nature of the tail, it helps in sperm motility. So vibratile nature under the sperm movement takes place by the tail, the movement of the tail. So vibrating nature of the tail, so it helps in the sperm motility. So this is a tail region. Okay, understood? If there is any doubts you can ask me, otherwise we'll go to the next concept. Next concept means oogenesis process. Oogenesis is the structure of egg. Okay, structure of sperm we have studied and structure of egg also we have to study. That is we are going to discuss in the next class. This is enough today, and in the next class we are going to study the structure of egg. Otherwise in the second chapter also the same thing you are going to study, that is human development is there. In that human development again you have to study the structure of egg and the sperm. So sperm already you are studying here, and there is no need to study there. And the development process you have to study in the developmental biology. There is a process from egg to an adult. So that in that time we will discuss the structure of ovum. Okay, thank you. We'll end up this session.